turnout. Thanks. So this is the PhD thesis defense for Brandon, who actually is my first PhD student coming here. So I've been fortunate enough to be his chair. Other committee members are Karen, Mike Varner, Josh Schiffman, and Guilherme Delfio. And it's been a real pleasure working with Brandon. He has a fire in his belly and has great ideas and has been really, really productive, I think, and I think has made some great contributions to the field. So I think what we'll do is have Brandon give his talk, and let's hold questions until the end of the talk, and then we'll go from there. So take it away, Brandon.
Cool. Awesome. Thanks. I've got a number of slides, and given the time constraints, I'm going to fly pretty quick. And so hopefully if I go over stuff really quick, just wait till the end and I can help clarify anything that doesn't make sense. But I'm going to try to go quick. All right, so my thesis is on clinical decision support for whole genome sequence information. A little bit of background so you know where I'm coming from. My background is in biology. I did a degree in human genetics, and I've worked on personalized healthcare in different areas. I've worked in the private industry. I've worked for the government in areas related to personalized healthcare and genetics. And then I came back to do my PhD, and this is all work related to things I've been doing. But most importantly, what gives me the credentials to talk about what I'm talking about is that I used to be the mascot Swoop here when I was an undergrad. And so if anything else, this is the most important thing, and it allows me to talk about what I'm talking about today. So a little bit of background. Personalized medicine. This is an area of interest to me. And personalized medicine is defined as the tailoring of medical treatment to the individual characteristics of each patient. And so what are these characteristics? Well, it can be anything from phenotype, medical history, social history, environment, family history, genetic information, biomarkers. All these things can personalize treatment and care to an individual. And personalized medicine is all about using this information to personalize that care. One of the areas that I see in personalized healthcare that's an issue is that we have an area of personalized medicine research, and I call this the promise of personalized medicine. This is where people are saying, with personalized medicine, we can do this, and we can, if we study this, we can achieve personalized medicine. And very much, this is the bench. This is the research. This is what people are out there trying to prove, and this is where a lot of money and effort is going. But then we have the realization of personalized medicine. This is where personalized medicine actually has an impact, where it's improving care, increasing the safety, decreasing costs. And that's the realization. But there's a huge gap between the promise in the research and the actual application of personalized medicine. And this is largely due to a number of barriers that inhibit the promise becoming a realization. And typically there's a 17-year lag between translating research into medical practice. And we believe that this translation lag is going to be even longer because of personalized medicine, because it's more complicated and there's those specific barriers for personalized medicine. But clinical decision support is a way to overcome these barriers and allow the promise of personalized medicine to be realized. And this is really what I came back to do my PhD on, was I realized that decision support is going to be key to realizing personalized medicine, and that's largely what I focused on. And so decision support. Essentially what it is a way to, decision support provides clinicians, patients, and healthcare stakeholders with patient-specific information, so again, a personalized medicine, intelligent filter presented at appropriate times to enhance health and healthcare. So that's the definition of decision support. And examples of decision support, it can range anywhere from medication dosing support, order facilitators, alerts and reminders, expert systems, workflow support. So there's a lot of ways to support clinical decision-making at the time of care. And so when I first got here, I teamed up with Ken, and we did a systematic review of decision support in personalized medicine, and specifically around genetically guided personalized medicine, which we define as genetics, using genetic information, and family health history information. And so in the systematic review, we used two literature databases to identify the literature systematically, and we did a review using search terms based on previous systematic reviews on decision support, genetic health services, and family health history, and we combined these search terms into one to specifically identify articles related to decision support for genetically guided personalized medicine. And in this review, we found the initial assessment got about 3,500 articles, of which most of them were excluded because they didn't meet our criteria. 300 of them were reviewed to see if they met the eligibility of our study. And in the end, 38 articles related to decision support for personalized healthcare was included in our final analysis. And just really quick, 22 of them, the majority, were decision support for cancer-related management. Specifically, there's several around family history, a dozen. And this is an example of a family history decision support tool where it collects family history and then provides a recommendation. You can't see it, but it says this: we recommend that this patient be referred to a genetic counselor based on their family history. And then other genotype-driven decision support tools, and this is actually a breast cancer risk a lifetime risk assessment tool, and it includes genetic information among other types of information as well. So these are two examples that we've identified in literature that are decision support to support personalized healthcare. Other areas that we found are decision support from pharmacogenomics, and so this is an alert that around warfarin, and this is actually done here at the U. And the other one was on decision support for other types of genetically guided personalized medicine. And this is a tool that interprets variants within people's genes, Gene Insight Suite for those who are familiar. And so those are the type of decision support tools that we identified in that systematic review. And we also we did an analysis of the results of these papers, and we found these trends. And so there's an increased number of decision support literature on for personalized medicine. And specifically, the dark line right here represents decision support for family health history. The light line represents decision support for genetics information. And so we're seeing a trend, an increasing trend of decision support for genetics information. And over here is a trend showing that standalone decision support tools, which is what they were traditionally, but they're becoming more integrated with EHR systems. And so those are the type of trends that we identified, which are good trends to see. But one of the things we identified were that there were no publications on decision support around whole genome sequence information. And we thought that was quite interesting and alarming given the future of medicine involving whole genome sequence information. And so we felt that this was a problem that needed to be addressed. And so we put together a manuscript describing the need for decision support for whole genome sequence information, essentially justification for why we need this type of support. And as a summary, genetics historically has typically been around monogenetic single gene disorders. And this is something fairly manageable by clinicians, and typically genetic counselors would be able to handle this quite easily. However, today, as genetics capabilities are increasing, it's becoming more widespread, and everyday clinicians are starting to see genetics in the clinic, whether it's patients bringing in direct-to-consumer tests or just more testing available for different genetic tests. And it's starting to become overwhelming to everyday clinicians. But tomorrow, when whole genome sequence information is widely available and everybody's getting the whole genome sequence, it's going to overwhelm the healthcare industry, given what we have today to handle this information. If anybody sat through a genetics lecture, they've seen this slide a million times, but it's to show that the cost of sequencing a genome is dropping dramatically. In fact, it just came out a few weeks ago that the cost of sequencing a whole genome has hit that $1,000 mark. And so we're going to start seeing whole genome sequencing being used much more in the clinic. And in fact, it's already being used in the clinic. Here are a number of studies related to doctors and researchers using whole genome sequencing in the clinic to diagnose diseases and to help guide treatment. Now, these examples are for rare diseases, undiagnosed diseases, but it just goes to show that whole genome sequencing is already being used in the clinic, and it's just a matter of time before it starts creeping out from those rare diseases to more common diseases that are seen in the clinic. In fact, Francis Collins, the director of the NIH, has said nearly every human disease has a genetic component. So one could see that this whole genome sequence information could be collected and used anytime a clinical decision is made. So where are some specific examples of how whole genome sequencing can be used? One is around clinical diagnosis. We can, patient comes in, unsure of what the patient is going through. You can look at the gene, and that can help guide the decision-making process. A risk assessment. Some of us carry genes that predispose us to increased risk for various diseases. Whole genome sequencing can help us identify those risks in patients. Reproductive screening, genetic testing is used now to assess for carriers for various newborn diseases. Pharmacogenomics, using genes to guide treatment and therapy and therapeutics. And then nutrigenomics, how genetics can be used to guide dietary and nutritional decisions. So those are all areas of whole genome sequencing information can be used in a routine clinical care setting. But there's a problem. In a routine clinical care setting, this is what a genetic test result looks like. It's a PDF or a paper of genetic test results. And this is an example from ARUP, but most labs do it this way. And this is for a single genetic test. I believe it's a BRCA test. That's just for a single gene. But the problem with whole genome sequencing is that it's not just a single gene. It's more than that. In fact, we have 20,000 genes in the human genome. With that, we have millions of variants and hundreds of relevant ones. And so how would a doctor know which variants are important, which are not, and which ones to include in the decision-making process? And there's also changing variant interpretations. And so right here, this lab report might say that the variant is likely benign or unknown what the interpretation is. But future research will come out and say, well, actually, this is a pathogenic mutation. You need to do something about it. Paper reports can't support that changing interpretation. And furthermore, paper reports do not enable computer processing. And that's, in informatics, sure, you can do NLP on it, but it's a lot easier to have something in a structured data in a format that we can use for decision making. Furthermore, genetics is complex. No longer is it one gene, one protein, one disease. We're starting to discover that it's networks of diseases. This is an example of the complexity of genetics, where this is a disease and the number of genes associated with that disease, and then how many of those genes are shared with other diseases as well. And so it just goes to show that genetics is not just a simple path from gene to disease, but it's a complex network that needs to be taken into consideration. There's many possible interactions that go into this. Furthermore, there's a lot of variants within genes. And so you have the common ones, the single nucleotide polymorphisms, but then you've also got insertions, you've got deletions, you got repeats, you've got large movements of pseudogenes that cause mutations. And then there's also splice site mutations that can all impact the phenotype. Also, you've got regulatory regions, you've got post-transcription modification, you've got a lot of factors that go into how the gene impacts the phenotype and thus the clinical impact. So given that complexity, we also have so much to know. Doctors have so much to know. In fact, today there are already 3,000 genetic tests available in the clinic. There's thousands of variants per gene. And like I said, there's 20,000 genes. And so there's so much breadth to know within genetics. But clinicians typically receive very little training in medical school. Typically, doctors receive about 6 weeks of basic genetics training. And this is typically related to monogenetic diseases like cystic fibrosis or Tay-Sachs that are inherited in a monogenetic fashion. But that's not what whole genome sequencing is going to take to interpret. So clinicians understandably rate their understanding of genetics as fair to poor, and there's many studies to show that they really do have fair to poor understanding of genetics and how to interpret it in the clinic. And then continuing medical education genetics programs aimed at increasing patients' education has been shown to be insufficient to meet the demands of whole genome sequence information. So you say, well, what about genetic professionals? These people specialize in interpreting genetic information. Well, in the United States, there are only 3,000 genetic counselors, which is 1 per 100,000 people in the United States. Medical geneticists, there's even fewer. And studies show that it takes about 7 hours to prepare for a patient. And so if everybody in the world, in the United States got a whole genome sequencing today, it would take 239 years for our genetics professionals to interpret everybody's genetic information. Furthermore, this is a distribution of genetic counselors across the United States. There's an unequal distribution of genetic counselors and genetic professionals able to meet the demands of whole genome sequence information. And so if whole genome sequencing is done in this area, we want to have genetic counselors there or genetic professionals there to interpret it. So it's going to be left up to the everyday clinician to interpret this information. And so given the complexity of genetics information, the limited capacity of clinicians to interpret this information, and the limited availability of genetics professionals, this is a big problem, especially with whole genome sequencing just around the corner. And so the solution, of course, this is what I'm talking about, is decision support. And what that is to make the everyday clinician who's doing his job, meeting patients, and enable that everyday clinician with computerized decision support to interpret whole genome sequence information. And again, the definition, but it's all about taking that complexity, that huge tidal wave of genetic whole genome sequence information that's going to be coming to the healthcare, and making it manageable so the clinician can use it in an everyday clinical setting without having to do any huge increases in their education or expanding the professional base in genomics. And so when I'm talking about decision support, I'm not talking about PDF printouts or standalone decision support or stuff that's emailed to the doctor alerting them of risks that a patient have. The decision support that I'm talking about is decision support that's integrated within the workflow of the clinician, within the EHR, at the point in time of decision making, with the recommendation on what to do with this patient and this genetic information. And if we do this, and it's provided automatically within the workflow, it has the greatest chance of being effective. And that's the decision support, the type of decision support that I'm going after. Furthermore, it's not just going to be good enough just to tell the doctor what type of variant a patient has and which gene. And so, who knows what APC gene is? Probably a handful of people. This is a gene related to colon cancer, right? So you want to be more explicit and say this patient is a high risk for colon cancer because of pathogenic mutation, and provide the recommendation with it, provide a recommendation for 2 years. But I would say it could be even better. All the clinician needs to know is this person needs a colonoscopy. And if they want to know why, they can click on it and get that information. But at the end of the day, the clinician who's treating the patient just needs to know what to do. And if they want to know why, they can dig in and get it. And it's important to point out, we'll get to this later, but this is decision support knowledge. The person needs a colonoscopy. That up here is genome variant knowledge. And that's going to, I'll explain that difference later when I describe the architecture. Here's an example of pharmacogenomics decision support. This is actually a system that is developed at Vanderbilt to interpret the patient's genotype and then provide patient-specific recommendations and alerts about this patient. And here, this person's at risk for an inadequate response to clopidogrel, and so they recommend changing that to a different medication. So that's an example of decision support within the workflow. This pops up when a patient orders, a doctor orders clopidogrel, and it's contraindicated based on the genetics information. But there's challenges to the goal. Genetics is still complicated, so we have to manage that complexity somehow. Our knowledge is still evolving. What we know today about the genome and how to manage the genetic information in the clinic is going to change 1 year, 2 years, 5 years from now. And so we have to have an architecture that's able to support this changing knowledge base. The current decision support infrastructure is unlikely to work on whole genome sequence information, given the complexity and the breadth of it. Whole genome decision support for whole genome sequencing will need to be scalable. It's not going to be sufficient for a single organization to manage all the knowledge around and the entire genome. It's going to require sharing knowledge between institutions. And so a guide to achieving this was developed by Dr. Masys as a technical desiderata for incorporating genetics information to the EHR. And he came up with 7 requirements. First of all, we have to separate the clinical interpretation from the genetic information, because clinical interpretation of genetic information can change over time, and so there must be a separation that maintained so that that can evolve over time. Secondly, when genetic information comes off the sequencing machine, it's large terabytes of data, way more than can be processed for decision support. So we need to have a way to condense this information to simple subsets that can be efficiently used to provide decision support and manageable in a clinical setting. Third, we need to maintain linkage back to the laboratory methods that generate the genetics information, because genetics results can change based on the machines and the type of laboratory methods used to generate them. Fourth, there need to be a clinically actionable subset. So there's a lot of variants and a lot of genes in the genome that just don't have any clinical meaning. And so it's important to separate those out from those who do have clinical meaning to make it easier for doctors to use it in a clinical setting. Fifth is to support human-readable formats so you can look at it, as well as machine-readable formats, so that health IT systems can process it and can make meaningful use out of it, and specifically for decision support rules. And so this is the first one that talks anything about decision support. 6, anticipate fundamental changes in our understanding of genetics. When the DNA was first discovered 60 years ago, what we thought about the genome is vastly different from what we understand today. Even 10 years ago, when the whole genome was sequenced, we thought there were about 100,000 genes. Today, we know that there's closer to 20,000. And that was just 10 years ago. Imagine how much our knowledge is going to change in the next 5, 10, 20, 30 years. And so we have to anticipate fundamental changes and be able to respond to those. And then finally, this genetics information needs to support both clinical care as well as discovery science. We're going to need to take genetics information, all the clinical information out there, and put it together so that we can make meaningful use out of our understanding and interpretation of genetics. And so of all these requirements, only one of them talked about decision support. And so as we're looking at developing a decision support architecture, we were going off of this, but we realized that there was a gap in the literature, that we needed a desiderata specifically around decision support, because we identified requirements that weren't addressed in the Masys desiderata. So what we did was we developed additional desiderata to augment that original desiderata specifically for decision support. And the additional desiderata that we developed, added on to the previous ones, is that decision support knowledge must be able to incorporate multiple genes and clinical information to provide the recommendation. And so it's not going to be sufficient to develop a decision support rule just based on one gene. We're going to have to incorporate multiple genes and multiple clinical information all into one to provide that recommendation. The next is that not only do we have to keep the variant interpretation separate from the genetics information, but we also have to keep the decision support knowledge separate from the variant interpretation and the genetics information, because this can evolve over time and so can this. And so these two have to be maintained separately to allow the evolution of knowledge. We must be able to support multiple EHR platforms with various data representations, largely because no one organization will be able to manage all the decision support for the whole genome sequence. And so it's going to require decision support deployed across multiple institutions, which the nature of the beast is that multiple institutions have multiple EHRs. And so we're going to have a decision support infrastructure that can support this. Next is to support a large number of gene variants while keeping the CDS knowledge as simple as possible. So this is an example of the CFTR gene, which is a gene associated with cystic fibrosis. And within this one gene, there are nearly 2,000 variants. Not all of them are pathogenic. Some are pathogenic, some are likely pathogenic, some are unknown, some are benign. So with these 2,000 variants, it's not going to be efficient to develop decision support rules around every single one of these variants. Rather, we'll have to group these variants into common clinical outcomes and then build decision support around that. The next is to leverage current and developing infrastructure and standards. And this is largely because genetics is complicated, decision support is complicated, and it's complicated enough, and to develop our own structure and our own infrastructure is going to be more work than it's worth. And so leverage standards and infrastructure that's already there to make it more simple to achieve the goals of decision support. Next is to support CDS knowledge base developed and maintained by multiple independent organizations. And so in this context, you allow specific knowledge authoring entities to expertly develop CDS knowledge and maintain it. So you can get warfarinedosing.com, and they completely focus on decision support rules around warfarin. period. And that's all I focus on, and you can tap into that and use it for your decision support. And then if you want to go to get oncology risk guidelines, you can go to someplace like ASCO, and they'll provide CDS knowledge around oncology risk guidelines. And so it's going to take this cooperation of multiple independent organizations to maintain the CDS knowledge base. Next is to access and transmit only genetic information necessary for decision support. And this largely is around security and efficiency. This is an example. This paper describes these researchers were able to take anonymous genomes that were publicly available, and they were able to re-identify these anonymous genomes based on markers within the Y chromosome and common surnames in genealogy databases. And so even though these genomes are anonymous, they were still able to identify the people whose genomes they belong to. And so we can't make entire genomes available for decision support because it can be compromising for security. And also HIPAA requires that only minimal information necessary for the actual clinical use case can be transmitted or accessed for that purpose. So what we did, we developed these. We had a core group of panels, a core group of individuals to serve on this panel. We developed these requirements, and then we distributed these requirements to a number of decision support and genomics experts as a survey. And as a Likert five-point Likert scale to assess the importance of each of these requirements. And each requirement, the respondents had an opportunity to put comments in below. And these are the results. So half of the respondents had expertise in decision support only, a quarter had expertise in the genome only, and about a quarter had expertise in both decision support and the genome. So those are the reported expertise of the respondents. And then these are the results. And so the light bar here is the desiderata was very important, and then it goes down from there, and the darkest is very unimportant. And we plotted this on this graph to show the relative importance. And as you can see, these desiderata were all judged to be importance by the people we distributed the survey to. And across the scale, the average p-value was quite low. So we can ascertain that these requirements are important, and we've essentially validated them among the community of experts. And so this was written up in a paper, and it's currently under review. We're waiting to hear back the final response on that. And so together, both the Masys desiderata and the current desiderata that we just developed provides the guidance to health IT developers to develop decision support capabilities for whole genome sequence information. So with that, now we've got that guidance. Now it's time to actually propose an architecture that can meet these requirements to provide decision support for whole genome sequence information. And so what we've proposed is to use service-oriented architecture to provide this decision support for whole genome sequence information. And just as a background, service-oriented architecture, COS CDS, essentially you have an EHR with clinical data, and that is sent to a decision support knowledge base, which is separate from the EHR. The decision support knowledge base interprets that clinical information based on their knowledge, and then it returns a decision support response, which is a recommendation, like this person needs a colonoscopy or a mammogram, or you should dose this much. And so that's the premise of SOA CDS. And that has several advantages. One is it's system agnostic. So it doesn't matter what the EHR system is down here, as long as you send it and receive it in a standard way, it doesn't matter what the EHR is. Next is it's scalable. So this one decision support knowledge base can potentially serve hundreds, thousands of potential decision support requestors or EHRs. And then finally, it's aligned with meaningful use stage 2. So the health IT requirements, it's currently in meaningful use stage, or it's planned to be in meaningful use stage 2, and there are efforts underway to make sure this happens. But nevertheless, EHRs are already starting to build in this type of capability. Epic, for example, is going to be coming out with, in the next 2015 release, it's going to be able to support SOA CDS. And so we're aligning with these capabilities that are becoming more and more available in EHRs. And so this is a typical, service request from EHR to CS knowledge base and back. Simple point-to-point, go back and forth. Given the complexity of whole genome sequence information, ours is a little bit more complicated. But and I'll go through the specifics. So one, we have the genome sequencing and annotation pipeline. So what happens is that the genetic information as a biological sample comes and it gets sequenced by a number of these sequencing technologies, and it essentially converts biological code into digital code that we can use for further processing. We've got to compress this data from the terabytes of size that it comes right off the machine to something that's more manageable, and this is done using a reference genome. So all of our genomes, we all have differences, but largely we're 99% the same. And so what this essentially does is it uses a reference genome and then looks at the differences against that reference genome. And that's the differences, or the variants, are what is actually captured in that file. And then finally, we need to annotate the location of variants that are identified and their relation to gene, their clinical impact, and location. And this is important because this really facilitates the rest of the capabilities around the genetics information, because it's not going to be feasible for decision support to go in and look through the whole genome at the raw data. We need to be able to pull out the specifics that we're looking at. So next is the database, where this genetic information is stored and how it's stored. And so the approach that we take is that we have a full variant database. So all the variants that a person has is stored in a full variant database. However, that's not made accessible to decision support. We have a secondary database, which contains the variants that are known to have clinical relevance. And that way, that promotes efficiency, because you're not searching the entire genome and all the variants. You're searching only ones that are known to have variants. And then it increases security and privacy. So you're limiting the genetic information available to outside queries. And then we're able to access the data through standard APIs. But then, of course, the databases can be connected. So as new information about variants comes out and they become clinically relevant, they can be pushed into a clinical genome database. And there could be managing entities that can support these genome variant databases. Laboratories like ARUP could take this role of managing this genetic data. Healthcare organizations can decide that they want to manage this genetic information. Or private companies, they could be even patient-controlled, or health information exchanges. So there's a number of different entities that can manage this. But what we're focused on is the actual architecture that can do this. Next is a genome variant knowledge base. The genome variant knowledge base plays an important role of updating our knowledge around a specific variant. So we can have a variant in a gene, but what does it actually mean? So this is a case where an APC gene, this variant, is known to be pathogenic because it's seen in other people. And those people who have it get the disease. So it's pathogenic. And there are large databases that actually maintain this knowledge around variants. HGMD is a common one that laboratories subscribe to that has a large, vast knowledge base of variant interpretations. Lovit is an open source genome variant manager. I put Myriad up here. They are a testing lab, but as testing becomes less and less laboratory focused, they become a knowledge base. And so Myriad has millions of has done millions of BRCA tests, and so they have millions of variant knowledge that they've built up, probably the largest knowledge base around BRCA results available. And so they can provide this knowledge on the variants. And they're a company, they'll probably sell it, but it just goes to show that these laboratories manage this variant knowledge base. And most laboratories have their own knowledge base as well. And then I put ClinVar up here. ClinVar is a publicly available knowledge repository supported by the National Library of Medicine. And so ClinVar is going to become essentially the central location for all these other areas to deposit their variant knowledge interpretations into. And I see ClinVar becoming, with more work that's currently underway, becoming that go-to resource for variant knowledge. Next, I'm going to talk about electronic health records. An important thing about electronic health records in this architecture is that it needs to maintain current or mandated EHR capabilities. So what that means is that we we can't, so what that means is that it stores clinical information, which it currently does. So it's currently available. It does not store genetic information. So we're not going to go to the EHRs and ask them to store genetic information because that's going to be done by that database that I just mentioned. And most EHRs don't store genetic information, so we're not asking them to do much. We're going to leverage the service-based decision support capabilities. Some EHRs already support this. Some are planning to. Eventually, if meaningful use requirements are accepted, all of them will support these service-based capabilities. Awesome. So, coming around the bend. So, these capabilities are going to be in EHRs. And then leverage the native decision support trigger and presentation capabilities of EHRs. And so what this means, the advantage is that it allows a consistent distribution across EHR vendors if we use the currently available EHR capabilities. So we're not requiring EHRs to build new capabilities that they're not planning to because if anybody's ever tried to get them to build anything, it's not very easy to get EHRs to change. And so we're not dependent on EHR development timelines. We're just using what's available. I think that's a key point. And then adoption coincides with CDS service capabilities. And so, again, as meaningful use accepts these capabilities, the adoption of this entire architecture capability will be supported. The next is the decision support knowledge base. And so, like I mentioned a little bit earlier in the Desiderata, it should be able to support multiple independently managed knowledge bases. And these knowledge bases can be free, so you just kind of I describe it like an app store, but it's essentially you can download a free app, you can use it, or you can pay for it. And some knowledge authors will want to develop knowledge and then sell that knowledge and make it available as decision support. And as long as there's standardized input and output and standards used to access that data and then return the results, this type of ecosystem, the App Store ecosystem, can be possible. And there are people out there developing these type of capabilities already. And so again, you can have specialized knowledge altering entities. So even you and researchers could develop specific CDS knowledge around their specific domain of knowledge, and they can maintain that. And the advantage is that it's expertly managed. And so you get warfarin dosing expertly managing knowledge around warfarin. You get the USPSTF expertly managing knowledge around all the guidelines that they've developed. And it's independently controlled. And so you're not able to control what other people do. You use the knowledge that they've made available. And the nice thing is that in a SOA CDS architecture, you can update it once and it updates everywhere because you're sending data to it and you're receiving data back. You don't have to go to each installation and update it. And then it also supports competition. And so if you don't like what warfarin dosing is doing, you can go to another service and subscribe to them if you think that they're better. And then the next component is the genome viewer, and this is important for the manual interpretation of genetics information. So this is an example of Amisha, which we use here at the U, where you can go in and manually review the variants, because some doctors and clinicians will want to go in and actually see those variants. And so we have to make that available. And so how this works, first of all, you have the genome variant knowledge base up here with that knowledge, and what this does is it updates the knowledge in the clinical genome database. This is important because variant knowledge in that database can change over time. And so it's important that the genome variant database has the most up-to-date variant interpretation knowledge available. The next step is that a certain trigger within the EHR allows the clinical data to go from the EHR to a clinical decision support controller. And the CDS controller essentially takes in that clinical information that's received from the EHR and it checks it against the requirements of the CDS knowledge. And it says, if the CDS knowledge requires genetic information, it checks for genetics information. If it's not there, it sends a request to the clinical genome database. The clinical genome database returns the genetic data that it's requesting, and then the CDS controller packages it together. So now you have the clinical data from the EHR and the genome data from the genome data, packages it together, and sends that complete data package up to the CDS knowledge base. At that time, the CDS knowledge base is able to process it and then return the CDS response to the EHR where it's presented as decision support. And so that's essentially how this entire architecture works. And there are several advantages for this architecture. One is each component is independently managed, and so you're not entirely dependent on other, on keeping everything up to date. And so you can rely on ClinVar to manage interpretations. You can rely on warfarin dosing to manage the CS knowledge, and you only have to focus on the specific aspects that you control. It leverages available infrastructure, so we're not requiring EHRs to build anything new. We're leveraging the variant knowledge bases that are already out there, and so we're using infrastructure that's already there. It aligns with a lot of the meaningful use requirements, specifically the SOA requirements for CDS, and it's scalable. So, and this is important because no one organization is going to be able to handle all genetics information, all decision support for whole genome sequence information. And so we have to have the ability to scale the capabilities across institutions, and this would support that. And so the question you might be asking is this actually going to work? There's a lot of moving parts. And so that's what we set out to do. And I teamed up with Salvador, he's originally from Mexico. He's a PhD student in England at the University of Sussex, and he was actually he was a visiting fellow with Dr. Kawamoto last year to work on SOA for healthcare and specifically around decision support. And my architecture was all around SOA. He was all about SOA and didn't really have a clinical use case and said, hey, let's team up. Let's build something together that we can achieve this. And so what we did is we built this architecture using these various components. I'm going to go into the specifics right now. So the first thing we did, we had to get genetics information, whole genome sequence information to use. And we actually used the 10-gen data set that was available through OMISHA. And the 10-gen data set represents the first 10 whole genome sequences that were made publicly available. And so if you, Craig Venter's on here, James Watson's on there. And so we were playing with their genomes and making decision support around it. So, and the OMISHA also provided the annotation pipeline. So it essentially said, all right, we've got the variants, and it assigned the variants to the genes and annotated it all. What we had to do, though, is build a clinical genome database. And so we had to take the genome data that was in OMISHA and make a clinical genome database that we could use for the architecture. So we built a MySQL, we just created a MySQL table using HeidiSQL, and we imported these genomes into this database. And we just created our own columns. And then we had to expose this database, this clinical genome database, to web services. And so working with Salvador, we created a web service interface to access that genetics information that was in the database. We didn't allow direct access to the database. So building this web service around it gave us the ability to access it for this architecture. The next thing we had to do was the genome variant knowledge. We used it from ClinVar. And so, again, it's publicly available, vast information, vast knowledge around variant interpretations. However, ClinVar in its current state isn't able to support our architecture. So what we did was essentially we pulled off the data set from the website, and it comes in this really huge, ugly XML file that's 610 megabytes, and it contains a lot of data that we just don't really need or care about. And we essentially had to strip that down and pull out the data that we cared about, and then we created a second table in MySQL to store the genome variant interpretation knowledge. And so this is data we pulled out from ClinVar in a table simplified so we can use it for our architecture. And so you have this gene with this RefSeq or variant, and then the clinical interpretation. And so that was the key part of that genome variant interpretation knowledge base that we needed to get access to. And then what we had to do is we had the patient genomes, so you have the gene and the variant, but it didn't have the interpretations, and we needed to update that information with the information that was in the data from ClinVar. And so we essentially created a script that matched up the two, and for those where it did match up, it provided the interpretation for that variant. So it's just it's a demonstration that yes, we can use data from ClinVar to update the variant, the patient gene variant interpretations. Of note, though, it took over 50 minutes for that query to run because there was so much genetic data and so much variant knowledge, and I had to go through all that. So it's a pretty big process to do that, computationally wise. And then the next thing, this is largely building on the work that Salvador does around CDS, COS CDS, and orchestration of services. And we specifically built it for the genomics use case. And so what this does, the CDS controller, it receives the clinical data in the form of a VMR and checks to see if the genetics information is complete. If it's not, it sends a request to the genome database, requests the data comes back from the genome database, and it integrates it into the VMR, and then it goes on to the next step, which is send it OpenCDS. We used the standards that we used the HL7 decision support standard to communicate the decision support requests. And then specifically, we used the VMR to represent the data that we were transmitting. And this is the actual observation result of how we represent the genetics information. So this is ABCA1 gene. We represent it as an observation focus. We included the observation value, but we don't actually build decision support around this. I included it just to show that we could. And then the interpretation is pathogenic, and that's represented using LOINC in this model. And so I created a simple use case of a 32-year-old male with no clinical history of cancer who does have a pathogenic mutation in the MUT-YH gene, which is a gene associated with hereditary colon cancer. And in this clinical scenario, whole genome sequencing was previously conducted, not for this use case, but it was just previously conducted. And our decision support was essentially accessing the information. What we had to do, though, is there was no mutation in the publicly available genomes that we used. There was no MUT-YH variant, pathogenic variant. So we actually had to create a variant. And I believe we actually used Craig Venter's genome. So our version of Craig Venter's got a pathogenic mutation from MUT-YH. And so what we did, we went to OpenCDS and we wrote the decision support knowledge to say, so these are the guidelines for screening recommendations for hereditary colon cancer genes. And so the MUT-YH has a biannual colonoscopy screening frequency starting at age 20. And so we created a rule that says patient is greater than 20, has a pathogenic variant in the MUT-YH gene, and hasn't had a colonoscopy in the past 6 months, then recommend a colonoscopy. So we created this simple rule, and we used Tolvin, enter the patient's information, and that's it. That's the decision support result. We show that this architecture can work, and that when we did enter this information, we had everything set up. Lo and behold, it does provide a decision support recommendation. This was a prototype just to demonstrate that it is feasible to do this, and we demonstrated that we've got that decision support result. And the architecture I described previously was built and functioned the way we described it for this use case. And so again, these are the components that we used. One of the things that we did was to do some performance testing on the various components to see how well it performed. And so we used SOAP UI to do these service-based performance testing. And we used how long it would take if you had 100 simultaneous users accessing these components. And so the first component we looked at was a genome app, which included the database and that service interface. And with 100 simultaneous users, we found that the average speed was about 40 milliseconds, which is about 0.04 seconds. That's how long it took to access the genome database and get the results back. So it's quick. And if anybody's watched the Olympics, especially if you've watched women's skeleton, you know how short of a time 0.04 seconds is. That's how long the girl who came in fourth place missed a bronze medal by. And then OpenCDS, we did the same testing there, and we found that the average speed was about 12 milliseconds, which is about 0.012 seconds. And for perspective on how fast this really is, when you blink an eye, that's about 300 to 400 milliseconds. And so these service requests are happening 10 times faster than the blink of an eye, which is awesome. But again, it was only 100 simultaneous users. What's going to be how is it going to impact if you have 1,000 or 10,000 simultaneous users? And so those are further testing that we would have to do. But there's still approaches that can be used to manage that performance. And then we looked at the overall speed, and we only used a user load of 20 simultaneous users because we found that the machines that we were using wasn't powerful enough to provide it fast enough when you got above 40 simultaneous users. And it's slowed down dramatically because of a process that uses to queue results when it's not fast enough. But that could be overcome if you have a faster computer. We just didn't have that. So we kept it to 20 simultaneous users, which is quite a bit. And we found that the average speed from end to end is 944 milliseconds, which is just under a second, which for those of us in the decision support world is a key number, because one of the 10 commandments of effective decision support is, well, actually, the first commandment is speed is everything. And they specifically say their goal is to reach sub-second decision support speed. And so we can show that this architecture, it's service-based, and it's able to access genetic information separately from an EHR, is still able to perform at that level. Now, again, there's still work that can be done to look at different scenarios, make more complex queries, look at more than one genome request from genome database, but multiple, and make the decision support more complex, which can all impact this. But it just goes to show that we're not talking about 20 or 30 seconds. We're talking sub-second. And so we were encouraged by the speed that we saw here. And this is written up in a manuscript that we are planning to submit pretty soon. It's done. We just got to review it and send it off. And so just as a summary, these are the publications that have come out of this work, starting with a systematic review at the beginning, and then this is the most recent one that we still have to find a home for. And 2 of them have been published, 2 of them are currently under review, and they've already received their initial review, and we've made changes, and so we're waiting for the final, essentially, acceptance of both of those. And so, so future direction, where are we going to go in the future? So there's a lot of stuff that we could do with this architecture. One is to incorporate multiple knowledge bases into this architecture, not just ClinVar, but also support the capabilities of ClinVar and expand those service capabilities. Develop the CDS knowledge base further. There's a lot of rules that could be developed around just everyday genetics, decision support, and knowledge that can be developed, but then also to improve tools to help others build knowledge bases for these areas. A big thing is to implement this architecture in an actual clinical setting, and then to show that there's cross-platform decision support capabilities across EHRs. And then, of course, improving performance, like I just mentioned, and then adding additional functionality. One of the things that we could have included but didn't in the CDS controller is the ability to incorporate prior authorization. And so some genetic tests requires somebody to manually review and authorize the genetic test. And that could be, you can include this into the CDS controller where you send a request to some type of prior authorization entity to process that. But you can do you can incorporate those type of capabilities into the CDS controller, even including a family history database. This is an area I'm interested in. If we had the clinical genome database, but also a family history database to incorporate into the rule, that can easily be added right here, and the CDS controller would check both databases and then send that information to the CDS knowledge base. The next is to build a genome repository that could be used, an open source genome repository that could be used, that could support both the research and clinical care. There's some genome repositories out there that support just research, but it'd be nice to have one that supports research and the clinical aspect I'm describing here. And then develop these for specific use cases and for specific institutions. And then look at doing large-scale implementations. And then one of the big things is to push for whole genome sequencing in a CLIA lab for research. So genetic testing is done for research. Push for it to be done in a CLIA-approved lab so that you can reuse that genetic information from research in this database for clinical care as well. So you don't have to resequence a patient's genome. So why is this important? This is actually a slide by Dr. Green, who's the director of the NHGRI, the Human Genetics Research Institute at NIH. And this is a slide that shows the the area of work and research and funding around genetics. And in the 90s, a lot of the work was just around understanding the structure of the genome and just barely getting into how the genomes interact with impact biology. And then over the past decade has been focused on understanding biology of genomes and its impact on disease, and we're slowly starting to creep out. Currently, we're in this phase where understanding the biology of disease, advancing the science of medicine, but it's still pretty early. But then beyond 2020, this is where the future of, where Dr. Green sees the future of genetics research going in applying the genetics information to clinical care to improve clinical outcomes. And that is largely the area of this work. And this is true. We did that systematic review. There wasn't very much out there on decision support. We're starting to see a lot more now. Even just the systematic review was published 2 years ago. It's already outdated because there's a lot more work that's been done on this area. But this is really where the future of research and where the future of NHGRI funding is going to be, is in applying genomics to improve outcomes of clinical care. And so it's a good field to be in for anybody who's interested in trying to figure out where they want to go with their research. And so that is the conclusion. I just have some credits. I'd like to thank the program Personalized Healthcare for putting food in my mouth and letting me hang out with them and getting me through my PhD program, and also the Department of Biomedical Informatics. Also my committee, especially Dr. Kawamoto, for guiding me through all this. He's been a great inspiration to teach me all the aspects of SOA decision support and included me. And really, a lot of the publications have come because of opportunities that have come to him that he's passed on to me that I was able to fulfill. And then others, Jen and then Salvador helped with the SOA prototype. And then, obviously, everybody in the BMI department. And, of course, my awesome family, my wife. And that's it. Thank you very much.